Hi everyone, this video is all about the first step of aerobic respiration, which is glycolysis. So we already filled out the top part of this foldable. So now you're going to move to this first little tab and write glycolysis. It's going to be on the next slide, so if you need to pause the video and write it later, that's totally fine. So what you're going to do now is lift up this top flat and you're going to put your glycolysis information right here. Okay, now there's only probably like 6, 7, 8 lines of text, so you don't have to write super small. Okay, just make sure that you can read it. Okay, so I'm going to stop this, stop the recorder, or I'm sorry, the video, so you have to look at me, and instead you can look at the PowerPoint. Let's see, it's not wanting to cooperate with me. There we go. Okay, so getting the PowerPoint up now. There we go. Okay, so you should be able to see this, although it's not showing me that you can't, so hold on, I'm really sorry, y'all. Okay, normally it highlights my screen if y'all can see it, and it's not doing that, so I'm hoping you guys can see this. Okay, so we are going to start by talking about the overall purpose of glycolysis, and the name actually gives it away. So glyco means sugar, and lysis, we've talked about that meaning like to burst or to break apart. So another word for break apart is to split, and so glycolysis literally means sugar splitting. So big picture, what's going to happen is we are going to have this 6-carbon sugar, okay, called glucose, the primary sugar that our cells like, and it is going to be split into 2 3-carbon molecules. And each of those 3-carbon molecules is called pyruvate. So we're going to have 2 of them, 2 3-carbon molecules. So we're literally splitting glucose into smaller pieces. So remember, y'all, that energy is stored in the bonds of molecules. So as glucose is broken down in like 6 or 7 steps, we're going to be extracting the energy from those bonds by capturing electrons in the process. Okay, and so before we dive into the details of glycolysis, let's talk about 2 important facts real quick. And that is that glycolysis happens in the cytoplasm of all cells, prokaryotes and eukaryotes. Okay. The other fun fact to remember is that this process occurs with or without oxygen, which is kind of surprising, right? Because this is aerobic respiration, which means it requires oxygen. However, this first step breaks the rules, and it can actually happen if oxygen is present or if oxygen is absent. But every step after this will require oxygen. Okay, so now let's talk about the process of glycolysis. So again, it's about 6 or 7 steps. Okay, you can divide those steps into 2 phases. We're going to have, let me change color here. We're going to have phase 1, which is the energy investment phase. And then the second half of glycolysis is called the energy payoff phase. So let's look at these terms, investment and payoff. So if I want to invest in a stock, maybe I want to invest in Cinemark, right? Because I'm hoping after the pandemic, people are going to go to the movies again. So if I want to invest in Cinemark, right, because I think they're going to make a lot of money one day, they're not just going to be like, oh, cool, Chelsea, thanks for having faith in us. Here, take some money, right? That's not how investing works. I have to put in money first, right? I pay into Cinemark, and then hopefully once they start making money, right, I start making money. And so the same thing happens in these cells. In order to start producing lots of energy, they have to invest a little energy first. So During the energy investment phase, which is the initial steps of glycolysis, the cell has to invest or use 2 molecules of ATP. So that's why there's a negative there, because the cell is losing that because they're putting it into the process. But luckily, by the time glycolysis is over, 4 molecules of ATP have been produced. So then what is the actual profit here in terms of ATP? What is the net yield? Well, you invested 2 and then you got 4 made. So you actually profit 2, right? 2 ATP. And that's usable energy that the cell can use. But it turns out we actually get a lot more from glycolysis. So I am going to write everything that we're going to get. So again, our starting molecule is going to be glucose, 1 molecule of glucose. And for every 1 molecule of glucose, by the time glycolysis is finished, we're going to have those 2 molecules of pyruvate that we already talked about. We're going to get those 2 molecules of usable ATP, but we're also going to get 2 molecules of stored energy in the form of NADH. So remember from the last video, NAD plus is an electron carrier, and it can accept 2 electrons. And so when it's accepted those electrons, right, it's become reduced because it's gained those 2 electrons. That's how we're storing energy, right? We're breaking down bonds and we're extracting the energy in the form of electrons. And NADH is holding on to those electrons. It's going to drop them off to the last step of this process. And so for right now, we call NADH stored energy. And this is a really important energy. This is actually going to help us mass produce ATP at the very end of respiration. Okay, so ATP is usable energy, NADH is stored energy, and pyruvate is also an energy yielding molecule because in the next part of respiration, we're going to continue breaking down pyruvate into smaller molecules and extracting energy during this process. Remember, respiration is a catabolic process. So we're breaking molecules down to release energy. Okay. And so the last thing that I want to introduce to y'all is how we actually made this ATP. Okay. So remember, y'all, ATP, and I want to change colors again. We'll just use red again. So remember, kind of leave a little space here, a little arrow. So ATP stands for adenosine triphosphate, meaning there's 3 phosphates. Okay, and that's what we're making. So what are we using to make ATP? Well, we're going to be using normally ADP, which is adenosine diphosphate, meaning it only has 2. Okay, and it's this 3rd bond over here on ATP, y'all. That's our high energy yielding bond. Because remember, phosphates have a negative charge. So 3 large negatively charged molecules don't want to be stuck next to each other because like charges repel. And so you can envision what's happening here with these 3 phosphate groups. Imagine this really big, thick spring that's coiled all the way up, right, against one side of the wall. When you release that spring, right, it's going to shoot out really fast, releasing a lot of energy. And that's kind of what happens here when we break this 3rd bond from ATP, which is what happens when the cell uses the ATP. Okay, so we need that 3rd phosphate group to have that high energy bond. So what we need during glycolysis, we have our ADP over here on the left, that's our substrate. And so we need to acquire this 3rd phosphate group from somewhere. And so it turns out during these steps of glycolysis, there's actually a molecule in the process that has a phosphate group. And so essentially what happens is an enzyme is going to cut that phosphate group off of the other molecule and stick it onto ADP. And so we call this process substrate level phosphorylation. So phosphorylation because we are adding a phosphate group to ADP, we're phosphorylating it, and substrate level because we stole it from a different substrate molecule. Okay. So we make ATP by substrate level phosphorylation during glycolysis. All right, y'all, that is the end of this video.